Hello guys, today we're going to be talking about the cell cycle. So what is the cell cycle, and what does it do? The cell cycle is a process in which a cell asexually produces more of its own kind, like skin cells for example. It's what distinguishes them from non-living stuff. In 1885, German physician Rudolf Virchow once said, "Where a cell exists, there must have been a pre-existing cell, just as the animal only arises from an animal, and the plant only from a plant." He had this concept summarized in a Latin phrase, omnis cellula e cellula, every cell from a cell. The genetic information of a cell, its DNA, is called the genome. The genome of a prokaryotic cell is pretty small, but the genetic makeup of a eukaryotic cell is enormous. To manage that much information, the DNA are wrapped up into chromosomes. Now, chromatin and chromosomes are the same thing, but they just vary in condensation. All species vary in chromosome number. Human somatic cells have 46 chromosomes, while our sex cells, or reproductive cells, have half that number. This was made so that when the egg cell and the sperm cell combine to form a zygote, we have one half chromosomes from the paternal side and the other half from the maternal side. This is what makes organisms that sexually reproduce more genetically diverse. Normally, before duplication, a chromatin looks like a long strand of chromatin fiber. As it duplicates, the sister chromatids will be connected by cohesins. This is a protein complex that attaches the two chromatids together. This type of attachment is called sister chromatid cohesion. The region in the chromatid that is attached more closely to the sister chromatid is called the centromere. This region's DNA is more tightly bound, giving it a narrow waist. The cell cycle is made up of 5 phases. You have the G1 phase, the S phase, and the G2 phase. Those 3 phases make up the interphase. The interphase is when the normal cell grows and develops. Then we have the mitotic phase, which is split into 2 phases, mitosis and cytokinesis. This is where the cell duplication starts. In 1882, a German anatomist, Walther Flemming, developed dyes for chromosomes. This helped him observe chromosomes during mitosis and cytokinesis. In fact, Fleming was the first one to use terms like mitosis and chromatin. In the G1 phase, the first gap, a cell grows and produces many of its cytoplasmic organelles, like the mitochondria or the endoplasmic reticulum. In the S phase, the cell grows more and also duplicates its chromosomes, but the chromosomes haven't condensed yet. In the G2 phase, the cell grows more and also produces many of its organelles that are crucial for asexual reproduction. When G1 phase and G2 phase were first discovered by scientists, they saw inactive cells, so they named them gaps. The mitotic phase is broken down into 2 phases, mitosis and cytokinesis. Now, mitosis is when the duplication actually starts. This is when the nucleus is split into 2 and the chromosomes go on each side of the cell. It's split into 5 parts: prophase, prometaphase, metaphase, anaphase, and telophase. We start with the G2 of interphase. Here we can see that the nuclear envelope is completely intact and the chromatin is still in jumbles. Here we can see that the centriole pairs haven't yet split. Now during prophase, the centriole pairs are split and they're moving towards the poles of the cell. We can also see that the chromosomes are more condensed now. There is an early mitotic spindle forming between the 2 centrioles, and the shorter spindles of the centriole that aren't in the mitotic spindle are called asters, which is just another word for stars. Also, just a note, in prophase, chromosomes are usually way longer than that, and they don't line up in straight rows. Alright, on to prometaphase. In prometaphase, the nuclear envelope begins to fragment, and some microtubules, the spindles, begin to attach to some of the chromosomes. These spindle fibers are called kinetochore microtubules because they connect to the kinetochore proteins on the chromosomes. The spindle fibers that don't connect to the chromosomes interact with the spindle fibers on the opposite pole. During metaphase, the centrosomes are now on the opposite sides of the cell, and the chromosomes are lined up in the middle. The chromosomes are lined up in a place called the metaphase plate. Usually the shortest of phases, anaphase separates the sister chromatids from each other. In this phase, the two daughter nuclei are formed, and the nuclear envelope is formed from the parent's remnant nuclear envelope. And any remnant spindle fiber is depolymerized. In cytokinesis, the organelles are split evenly across the cell, and a ring of actin forms a cleavage furrow.